It is a great moment of extreme pleasure to introduce our resource person, Mr. Prateek Kumar, for the last module of this workshop. Mr. Prateek Kumar is a PhD scholar at IIT Mandi. He has earned his master's degree in bioinformatics from Jamia Millia Islamia University, New Delhi. He did his bachelor's in zoology from University of Delhi, New Delhi. He currently works on different systems of proteins to study their folding, unfolding behavior, disordered property, and structure-based inhibitor designing. His ongoing research on these topics emphasize on various aspects such as molecular dynamics, simulation, virtual screening, protein-protein interaction and analysis. His work include understanding the protein dynamics in different environment, modulation upon interaction with lipids, and identification of inhibitors against specified targets. He has also published more than 35 national and international research articles in reputed journals and book chapters. So, may I invite our resource person, Mr. Prateek, to commence his hands-on training on dark proteome analysis. So, please welcome Mr. Prateek, sir.
Thank you, sir, for your kind introduction. And before going for the hands-on, I would like to thank all the organizers of this wonderful seminar and this workshop. And as Rajnisha said, there's so much to tell about the proteome. So yes, Rajnisha said that there is so much to tell about dark proteome and IDPs. So let me begin with the short presentation on this dark proteome perspective. So dark proteome perspective, basically I'm going to talk about these intrinsically disordered proteins based on their sequence and structure-based analysis of IDPs. So starting with this, IDPs or intrinsically disordered proteins, as previously told by Rajnisha, there is a very dark region in our proteomes, in eukaryotes, in viruses, in bacteria. Every species have the dark regions, which is not explored, which is not known, and something in the dark proteome there is called IDP, that is intrinsically disordered protein, which don't have any structure, which don't have any rigid structure of their own, but they perform many functions in spite their no structure availability. So there are so many things about dark proteome, which are some speculation, which are some experimental observations. So one study conducted in 2015 told that out of they conducted a survey on 546,000 proteins of from the UniProt. So they found that 44 to 55% of the proteome in eukaryotes and viruses was dark. So basically, that dark proteome means that those proteins don't have any structure, and they can perform the functions despite having no structure. So in comparison with the viruses and eukaryotes, archaea and bacteria only have 14% dark regions. So this can be comparable that based on this comparison, these eukaryotes and viruses have large region of dark proteins than the bacteria. So also another report said that 44% of genes in humans that code for proteins contain disordered regions. So these are the studies. There are so much of studies in last 2 decades, but now I'm just showing just these two. So I will give a gist of these disordered protein functions. These disordered proteins are unfolded or unstructured, you can say. So these proteins have so many functions, starting from the sequence. and they can be analyzed through the sequence. They also regulate the protein interaction and cell machinery also, and they are also a part of membraneless organelles also. So for they are lower in case of sequence. If we are talking about sequence, then this means that lower aliphatic, aromatic, and cysteine amino acid, and highly polar and charged amino acids are involved in IDP regions. Same case with PPI and regulation. Functions like coupled folding binding and allosteric regulation are are a function of IDPs. Ligand induced regulated unfolding are also function. And membraneless organelles are like causal bodies and nucleoli possess some IDPs in their organelles. So they are different method-based classification of predictors. So for using the sequence of these IDPs, we can predict their nature. Like, are they literally disorder or order, or they can function? They can perform any function or not. So the methods which are available online nowadays, these are neural network, meta predictor, physiochemical based predictors, and SVM, that is simple vector machine based vectors. IDP is a kind of protein, like they are unstructured. So, and morph is where you can see the morph. Morph is a molecular recognition features which are identified in the disordered proteins or disordered regions, which perform the functions like protein-protein interaction, protein nucleic acid interaction, and other functions as well. So these predictors, like neural network-based predictors, are Ponder VLXT, VSL1, VL3, this RDP bind, and meta predictors are Ponder fit. And similarly, physiochemical based properties like IUPAD and fold index and etc. And SVM based are PR DOS, MOCPAD. So I will be giving some hands-on on some of these predictors like Ponder and IUPAD and Anchor, PR DOS, and Ponder fit as a meta predictor. Before going for hands-on, so let's have a look about what the structure and unstructured and partially structured proteins. So I have shown here some currently ongoing this pandemic virus proteins. Like this one is the spike protein of SARS-CoV-2, which you can see that is a completely ordered or majorly ordered, but it's see the C-terminal end, as in the last presentation sir showed that it is a disordered region. So any protein can have disordered or ordered regions. So this is not the consensus that every protein is disordered or disordered. So this is the majorly ordered protein, like 90% ordered protein. It's around 1273 amino acid long, but and the around 1100 amino acids are ordered. This one is the structure of rotavirus RNA dependent RNA polymerase, the B1. Though this one is also majorly ordered. C is partially ordered and partially disordered. In case of it also have alpha beta chains. So this is a CDK2 of human. The D is the cyclin A complex, and E and F are NH2B, NH3 protease from Zika virus that Dr. Rajni sir was talking about in the last presentation. So these proteins are highly partially disordered and bound with a protein. Like this one is the another protein which shown with another color, and this one is the other protein. So there are two proteins combined with each other. It means that in presence of this protein, like I will say this one is the NH2B protein and this one is the NH3 protein. So in presence of this NH2B, this protein will only function, but and in absence of this blue or NH2B protein, this protein will not function. Although it is a disorder, but it has a function now. So you can say that the disorder proteins also have function. And this one is also a structure of SARS-CoV virus. This is 2CME, ORF8. Now you can see the highly disordered regions of a protein. If you're looking for this kind of threads, these are all structures are taken from NMR. These are short structures like 50 amino acids or 40 amino acids. So these are very disordered structures. You can see containing some alpha helix of around 10 to 12 residues, but majorly structures are disordered. This is the ORF9b protein of coronavirus. And this one is also such a human C2S2 type zinc finger protein, which is completely disordered. You can see this here. There is only a thread-like structures on nothing, no alpha or beta sheet there. Similarly, this is also a SARS-CoV-2 nucleic acid phosphoprotein N-terminal domain, and this one is the zinc-driven domain of human general transcription factor TF2B. So these are the structures which are ordered, partially disordered, and completely disordered or majorly disordered. So these are the common predictors based on different algorithms which we use or generally used for the analysis of disorder-based regions in a protein or in a polyprotein. So I will be, as I previously said, I will be discussing some of them. So we can have some little description here that Ponder VLXT, which is a free-forward neural network trained by the backpropagation algorithm. Similarly, a neural network based on BSL2, BL3, and one is PonderFit, which is a meta predictor that combines output from 6 predictors. Sorry. One is IUPRAD, which uses pairwise interaction energies for disorder prediction and also uses physicochemical properties of the amino acids. And similarly, there one is MobiDB, which is also a consensus-based predictor and aligns the manually curated databases. Manually curated databases contain an experimental as well as predicted databases and the prediction for disorder. PRDOS, which is a simple vector machine algorithm for position-specific search, and also uses PSI BLAST for homology search, and gives the output of conserved disorder regions in there. So here is a meta disorder machine learning based meta predictor, which gives the output from 13 other predictors. So I will be discussing about those later on in the hands-on session. There is one more popular predictors of disorder-based binding regions, which are morph regions or nucleic acid binding regions. Basically, these IDPs are binding with other proteins or other nucleic acid, any nucleic acid or any kind of molecules of like that. This can be transporting vehicles, anything, vesicles or anything. So these are the predictors which can be identified for the morph, which are the protein-protein interacting regions in the disordered regions or in the ordered regions. So anchor, morph-paired, morph-GB web, PSO-3. So these can be used. Similarly, there are softwares for DRNA PRAT, DISO-RDP bind, and PR print for analysis of nucleic acid binding regions in the structure. Now you can see I have shown some windows from the nucleic acid protein of SARS-CoV-2, which is the output file of Ponder, DISO-PROT, and IUPRAT2A, MobiDB, PRDOS. We will be using these predictors today for the hands-on. So now we have used these sequence-based analysis of the IDPs. Now we can also use MD simulations for IDP analysis, like structure-based analysis. For structure, because we know that IDP don't have any structure, or they have a partially or fully unstructured regions, like they have random coils majorly. So due to that, the structure of those regions cannot be deciphered through the X-ray, through the cryo-EM. So people are trying to develop some technology for identify these structures too, but currently there are no such technologies available. Therefore, we use MD simulations to identify the conformations of structure. So that a structure is modeled by any predictor or any software like iTesser, RaptorX, or AlphaFold now. So these tools are very highly used, and they also provide very reliable data and reliable structure for this. So using these structures, we perform the simulations. Simulations are basically used to investigate the structure, dynamics, and thermodynamics of biological molecules or chemical molecules or something polymers like that, and their complexes also. So we can also identify the protein stability, conformational changes, protein folding, ion transport, and several other problems in the real physiological conditions. So for virtual physiological conditions, and also we can perform drug designing using these simulations. So here's a comparison with the experiments and with the simulations or in silico work we can see. So in experiments, what we do, we invest so much of amount of money and resources, and still we get a macroscopic view, which is also a good thing. And using MD simulations or any computational work, we can get a microscopic view. Like we can get to know about the atoms and their rotation and their bonds and bond angles, bond stretch, bond strength, like those information about any protein. So currently, the structures, the simulations algorithms are available which can predict or calculate many local motions, rigid body motions, large scale motions to identify the atomic fluctuations and to folding and unfolding of a proper domain. So, according to a study or according to the force fields available, we have local motion. Like we can identify the local motion of any proteins, amino acid and the peptides. So within which can be in the distance of 0.0125 angstrom, having a with a time scale of 10 to the minus 15, that is a femto, and 10 to the minus 1 second. So in this reason, we can identify the atomic fluctuation, side chain motions, and loop motions. So and so on. We can, if we have to find out the helix motion or domain motions or subunit motion, how they are performing. So we can perform the simulation up to nanosecond to microsecond or milliseconds. Similarly, large scale motions, like if we have to decipher the dissociation and association of any molecule or any domain, or folding and unfolding of protein or domain movement like that, then we can go for a simulation up to from microsecond to second level. Nowadays, getting up to microsecond or nanosecond is enough, but people are trying to get to seconds also. So what is the relevance of molecular dynamics simulation with structural biology? So this is we can say that the MD simulations are capable of determining conformational dynamics, structure compositions, and organization of proteins in an aqueous environment. Similarly, we can also identify the interaction of protein with lipid molecules, inhibitors, with any other partner proteins, etc. Also, there are several packages available for simulations like Desmond, Gromex, Ember, NAMD. So they can be used for the simulations. You can see this. These are the two structures available for I have shown here, which are simulated for 1 microsecond and modeled with the difference of 7 residues at its end terminal. You can see that having a difference of 7 residues at an end terminal, and there's a lot of difference in the structure as well. This is fully disordered, and this is partially disordered. And they're also getting it is not getting any structure confirmation between during the simulation, but it is getting some structures, not fully structured, but some structures during the simulations. So what is the MD simulation? What upon these MD simulations work upon? So we can say that if any MD simulations that works with the force field. If force field can be explained as the interatomic potential energy of a system, which is calculated among several parameters such as bond of initial and final frames, and bond angles, torsion angles, improper angles, electrostatic and energies and charges also. So this can be the atomic coordinates which defines all these things: bonds, angles, and torsions, impropers, electrostatics. So during the simulation, when a frame changes, when the position of any coordinate changes, that coordinates provide these values, and the initial value remain the same. This r naught or theta naught remains the same, but this value gets changes. So based on this the difference, we get to know about the potential energy, and we can calculate the energy of the overall system. These atoms are held together by simple harmonic. You have also read in the your physics class in 12th, 11th. So the simple harmonic motion has elastic forces. So these are bound with those simple harmonic motions, elastic forces, which represent within the specified region for simulation. So it can only go for some specific regions from here to here, not like from here one corner to other corner, not this, but from one position to another position. This is fixed with a random or this angles. So there are so many force field available like OPLS, Gromos, Charm, Amber, Druid, which are optimized for this kind of structural of composition and investigating protein aggregation, binding efficacies, conformation changes, etc. These are the water models in protein simulation we use. Like for any simulation setup, we have to provide any like physiological condition. We provide some buffer conditions where we provide water, we provide ions, many things. So like that, we provide water models in the simulation setup, which are which can be SPC, TIP4P. These are the types which have been evolved during these 2 decades. So these SPCs is a sample point charge with 3 interaction sites. TIP3P is also a 3 interaction site. Like if they have the site to interact with the protein atoms from H, this is hydrogen, or oxygen. So it can interact with these three sites, having an angle of 109.47 degree. So similar, the SPC is little bit modified with and optimized with the advanced algorithms. So it will also have the same angle, same sites, interaction sites, but there's a difference in the angle with around 5-degree reduced angle, 104.5 degree. There's one more TIP4P, which also has a 104.5 degree angle and have a dummy atom to interact with any proteins, amino acid atoms, or anything. The 3-point charge has 3 interaction sites, 4-point charge has 4 interaction sites, and additional where the additional dummy atoms provide the interaction site, which is used to improve the electrostatic distribution, like this charge distribution all among the simulation setup. For analysis, what we use for when a simulation is completed, what we use for analysis like RMSD, RMSF, radius of gyration. So what is RMSD? RMSD is the root mean square deviation, which is measured between 2 sets of atoms. Like which one? One is the previous one. Previous one means the initial one, and the final one. That is for any frame you want to give, like first frame, second frame, 1,000 frame, something like that. So there's a difference between the distance of those atoms. And second thing is average of all simulation frames in this trajectory. which is known as root mean square fluctuation, RMSF. Third thing is radius of gyration, which states the compactness of a protein structure. So you can see a representation of RMSD for 1 microsecond data. Like there are thousands of frames, and every frame is fluctuating highly for an IDP. This kind of data is shown for IDPs only. While for any globular protein, which is a rigid structure, it will give a linear line, not a fluctuating line. So this is also gyration data, which is also plotted with the respect of time. It also has very fluctuating values, which can also confirm like that the structure is very unstable, structure is very disordered in that case. See, similarly, there is a value of shown value of RMSF with the residues, and those residues is fluctuating very high, which have a value of with a peak of 1 nanometer, like or 10 angstrom or 14 angstrom, like that. But these residues are fluctuating very less, so you can say that they have some property to attain some structure. Similarly, here there I am shown that there is a change of hydrogen bond also. At every time frame, number of hydrogen bonds changes, because if any structure gains any structure, any helical or beta sheet, it forms hydrogen bonds. So if in next frame if it is not forming any structure, so it is not getting any hydrogen bonds. So like that, for thousands of frames, we see this kind of graph. This is the secondary structure analysis shown here. That this yellow part shows the beta sheet. This part shows the turns. So mostly, and the white part shows there is no structure propensity, and this pink part shows the helical. So there is the analysis says that there is highly disordered regions available in the structure, and this is the free energy surface, which can be plotted with the all the frames energy. So with these RMSD and gyration values, we can say with those that any clusters of those structures with the same RMSD or with same gyration are falling in this region, and with other with the different RMSD in different gyration and higher energy, those are falling in this line. So you can say this IDPs are very fluctuating, but the residual structure is not fluctuating. And so like that, if you have any residual structure, there must be the clusters forming only one place, so there will be no changes at all. This is a replica exchange MD simulations, which is also done for the IDPs or IDPRs. This means for regions. So here I have shown some data from a paper we just published in a journal, that is shown that using a structure, the model structure, which I've shown here, this one structure, we perform the simulation, REMDs here. What is the REMD? REMD is the parallel simulations. I'm telling in some layman language. In parallel simulations, multiple simulation running in the parallel conditions with the same parameters with one structure in all replicas. Like we have here 8 replicas starting from 0 to 7, and we have given a single structure in all the replicas, but there is a change of temperature from 298 Kelvin to 410 Kelvin temperature. So what is done in these temperatures? Replicas. At every temperature, every replica swap their conformations from one to another. So at any places, any structure, if the structure can attain any conformation, like any rigid conformation or any beta sheet helical forming capability, so that can attain after getting from one temperature to another temperature. That means that was trapped in a local minima. So if we have to provide a global minima, then we have to perform REMD simulations. So here we have performed RM simulation. We found that in some at some higher temperature, the structure are forming some beta sheets here at 394, at 378, at 410 Kelvin. It is forming some structural conformations. So, what are the biological relevance of in silico predictions? Here, you can see that I have shown microscopic and microscopic differences in experiments and in silico predictions. So, in silico predictions are used to study and understand the concept of disorder by in silico methods, highlighting the need for drug designing, which targets the signaling hub in the proteins associated with many diseases. Like now we are having this COVID-19, and we have also shown for the first time our lab has shown the SARS-CoV-2 proteome disorder perspective and found that a protein with 410 amino acid is completely or is majorly disordered, like nucleic acid protein, which can be linked with its pathogenicity. And we are trying to work, we are also working on that. This characterization of the uncharacterized protein helps us to understand many unclear biological functions of the protein. In viruses, IDPs are targeted that hijack the most molecular systems and disrupt the host normal functioning. So these are the biological elements. And this the short presentation is over. And now I would like to thank my PhD guide, Dr. Rajneesh Giri, Biotech Center, IIT Mandi, SPC, the Science Cluster for Simulations, and IIT Mandi for facilities, and DST serve for funding. And this is the campus where we live, where we work, and where we publish studies. So thank you for this.